Welcome. My name is Claudia Melo, and I will be presenting the antioxidant, antibacterial, and anti-SARS-CoV activity of commercial products of Xylopia. Xylopia is a spice and medicinal plant that grows wild in many West African countries. The dried fruits are used as a flavoring for soups and traditionally in decoction as an analgesic and anti-inflammatory, as well as a treatment for infections. The medicinal properties of the fruits are associated with the presence of chemical compounds found in the fruits, such as phenolics and flavonoids, that gives the plant an ecological advantage. These chemicals, they are associated to many biological activities. The objective of this research is to study the antioxidant, antiviral, and antibacterial capacity of 5 different commercial samples of Xylopia fruits from Ghana and to determine the phenolic content using different solvents. The first thing that we did in figure 2a, we used a modified phenolytic assay that is used to test the concentration of phenolics using 6 different solvents. In this case, we used DMSO 80, 70, 60, 50% ethanol and decoction for 30 and 10 minutes. We observed that when we used boiling water, it consistently extracted the highest amount of phenolics in all of the samples, and this method is the one that we use for further experiments, while the ethanol solution extracted the amounts. Sample 4 and 5 showed the highest amount of phenolics, while sample 2 showed the least. We also look at the concentration of flavonoids, which are a subgroup of the phenolics, and observed similar results. I'm referring to figure 2b. Following, we tested the antioxidant activity of the extracts using 2 different tests, the ABTS and DPPH test. In the ABTS test, in which you can observe that is figure 2c, we observed that the highest antioxidant capacity was observed in sample 4 and 5 using that decoction method. In the DPPH test, we observed that there was no difference between the samples, and this tells us that the ABTS assay is more sensible and accurate than the DPPH. Observing these results, we hypothesized that due to the presence of phenolic compounds, the extract will have different biological activities, such as the antiviral and antibacterial. So we tested the antiviral activity of these extracts, and something to keep in mind when an antiviral test is performed is that viruses are obligated to insert the genetic code inside their host, in this case a cell, to be able to replicate. So keeping that in mind, we need to test to see if the extracts were toxic or not. So we did a dose response curve where we obtained a value known as CC50, or the half maximum concentration that is needed to kill 50% of the cells. Then we tested the antiviral activity of Xylopia against SARS-CoV-1, which is graph A, and the Xylopia extract against SARS-CoV-2, which is graph C. We did a dose response curve where we obtained a value known as EC50, or the half maximum concentration that is needed to inhibit 50% of the virus. So looking at these graphs, we can see that in the highest concentration of the extracts, we see a greater viral inhibition. However, as we dilute the extracts or we go to the left side of the graphs, we observe a greater viral replication. And with this curve, we get that EC50 value. The same thing for the CC50. However, in that case, we will look at cell viability. So with these values, we did a ratio between the CC50 and the EC50 value, where we obtain a therapeutic index or TI value. This value tells us if there is a true antiviral activity or not. If we see a value above 10, it means that there is a promising antiviral activity. For only sample 4 and 5, the TI were calculated and resulted in values between 1.6 and 3.1. These values suggest a moderate antiviral activity. Following, we evaluated the antiviral activity of chlorogenic acid, caffeic acid, and their combination, which we identified through HPLC and are known for their antiviral activity. Graph B is these compounds against SARS-CoV-1 and graph D against SARS-CoV-2. We found out that the antiviral activity does not justify for the moderate antiviral activity found in the extracts, and this suggests that there's other compounds that could be responsible for the moderate antiviral activity. Also, we studied the antibacterial properties, in which we built a growth curve using E. coli and Bacillus subtilis against sample 1. Bacterial growth was monitored hourly by spectrophotometry for a total of 7 hours. We observed that E. coli was only slightly suppressed by the presence of Xylopia extracts, and the growth of Bacillus subtilis was completely inhibited. So in conclusion, Xylopia extracts show antioxidant, antimicrobial, and moderate anti-SARS-CoV-1 and SARS-CoV-2 activities. And these results support the use of Xylopia fruits in traditional medicine. Future steps will look at to determine the antiviral activity against herpes simplex virus type 1 SARS-CoV-1 and explore other potential biological activities. Before I finish, I want to acknowledge the team that worked on this collaboration. I want to thank the Science Day Committee for allowing us to present this project. I want to acknowledge all the grants that funded this project, and specifically, I want to thank the Science Department of BSC for all their support. Thank you.